Hello, everybody. Welcome back to my YouTube channel. So this is the year of 2025. So today we'll be doing paper 3, which is bio paper 3. So what we're going to focus today is Visking tubing. So as you know, this is a very hot topic this year for paper 3. So I'm going to teach you the method of tying a Visking tube and also how to answer the paper. So we have a Visking tube here. I have a Visking tube here. You don't have a Visking tube, but I have a Visking tube. Visking tube, it looks like a plastic bag. It feels like a plastic bag. So it's gonna be a little bit hard when it's like dry. So you have to immerse into water. I have a video of how to tie a Visking tube. I posted it to my Wiling Biology Instagram. So please go there to watch the video, okay, how to tie a Visking tube. It's a very detailed way to tie a Visking tubing without leaking out, guys. The method is do not leak, right? So in the video, I teach you how to tie a Visking tube that is not leaking. So when you have your exam paper with you on the particular day, I'm going to teach you how to separate your time, how to manage your time when you're going for paper 3. Paper 3 is 45 minutes. So when you go in, you go and check. So they give you a list, right? Maybe they give you this kind of thing. They give you a list. So the first 5 minutes, you check the list first. Okay, and then when the experiment started, what you're going to do is, the next thing you're going to do is to put the Visking tube inside the beaker and immerse it, because I don't know whether they immerse it for you already or not, but you immerse it. Meanwhile, immersing for 5 minutes time, what you're going to do right now is you have to read the exam paper. You go through every single question. You write the answer that you know first. So you know the MVRE, right? You write down the answer first. Don't waste your 5 minutes. 5 minutes is a lot as well. Okay, you don't waste your 5 minutes. Okay, and you write your answer first. After 5 minutes, when the time is up for immersing of Visking tubing, then you start to tie the Visking tube. So the video, remember to go to my Wiling Biology Instagram to see. So this is my Instagram. Please go ahead and see. I posted there already. Please watch and you learn how to tie a Visking tube. So if you don't have a Visking tube, it's okay. Just use any plastic bag. You cut out on the end and use any thread to tie it. It's just a matter of tying. When you're able to do it, means you can have the very correct experiment without failing your experiment. So before we do any question, we're going to give you some of the theory that you should understand from the Visking tubing. So first, we have to know Visking tubing, Visking tubing, they are selectively permeable. Okay, selectively permeable. Okay, what does selectively permeable means? It means they will only allow certain substances to pass through. Certain referring to what? It means only small substances can pass through. Okay, what are the small things they can pass through? First, they can pass through, they can allow, okay, always okay, small thing, so they allow small thing to pass through. Small thing can pass through. Okay, the small thing here referring to glucose, referring to distilled water, referring to iodine. So there are three things that can diffuse across the Visking tubing. Okay, what are the things that cannot diffuse across? The one that cannot diffuse across is the large molecule. Large molecule cannot pass through the Visking tubing. Okay, what are the large molecule? We have starch, and we also have sucrose. These are the large molecule. They cannot diffuse across the selectively permeable membrane. Okay, so these are the things that can and cannot. So you have to know who can and who cannot. So what are the things that can still pass through? We also have glucose, fructose, galactose. They can also pass through selectively permeable membrane. They will only allow certain substances to pass through. Okay, let's say they give you two solutions. We have solution P and solution Q. For example, we have glucose and starch. Okay, to know about the concept, right? So let's say we start with glucose first. So we know that glucose, the original color is colorless. Colorless means no color. It's like water is called colorless. The original color of glucose is colorless. To do a test for glucose is called Benedict test. Benedict test is blue in color because Benedict test is copper 2 sulfate, so it's blue in color. So Benedict test is to test the presence of reducing sugar. Is to test the presence of reducing sugar. Okay, what is the reducing sugar here? The reducing sugar here referring to all monosaccharides and disaccharides besides sucrose. So we have glucose, fructose, galactose. Okay, this is the monosaccharides. We also have lactose and maltose. Okay, these are the 5 examples of reducing sugar. They are able to turn the blue solution in Benedict test, blue solution, into brick red precipitate. brick red precipitate. Blue into brick red precipitate. Remember, it's precipitate, not brick red. It's called brick red precipitate. Okay, the other one we have starch. So let's say for starch. Okay, starch original color is colorless. Either colorless also correct, or either you want to write cloudy color also correct. Cloudy solution also correct. Either both also correct. Either cloudy or either colorless solution. The test to test the presence of starch is called iodine test. Iodine test. Iodine test is to test the presence of starch. Okay, test the presence of starch. Iodine test original color is what color? Original color, it is yellowish brown color. Okay, yellowish brown color. They will turn into blue-black solution. You can write blue-black solution. You can write dark blue solution. But we cannot write blue solution. We cannot write black solution. It's either blue-black or dark blue solution. It's only two answers, either blue-black or dark blue solution. Yellowish brown, brownish, yellowish is up to you as long as it's yellowish brown or brownish yellowish. It's not yellow also, it's yellowish brown. So this is the basic that we have to know in this experiment. So we know Visking tube, we know glucose, we know starch. These are the three important things that you have to know. Okay, now again, in exam, the first 5 minutes for you to check your apparatus and material. Remember to check properly. Yeah. So they give you all of this. You're supposed to have all of these things. So they give you 5% of starch. They give you distilled water. They give you iodine. And they have stopwatch and glass drop. Okay, this is an experiment that you don't usually see in your exam because I forgot it's which book or which experiment. I forgot already. But it's the not the common one that you see, because the common one that you see, right, is something like that. It's something like that. It's what you see, right? But I'm going to give you something else which is slightly different, which is like that. They hang it with a rod. Okay, they say plasma membrane is a selectively permeable membrane that regulates the movement of molecule into and out of the cell. The movement of molecule through a selective permeable membrane depending on size of molecule. Carry out experiment to determine the effect of molecule size of the movement of substances across selectively permeable membrane. So we have diagram 1.1 shows the setup of apparatus for this experiment. So they give you a glass rod. We have Visking tubing, selectively permeable. Inside the Visking tubing, we have starch, 5% of starch suspension. Outside of the Visking tubing, inside the beaker, we have iodine solution and we have distilled water. How many percent doesn't matter because that will be a constant. So we have to look at whether they got starch, iodine, or distilled water. From what we understand just now, right? Starch is a large molecule. Can large molecule diffuse across the Visking tubing? The answer is cannot, right? They cannot diffuse across it, right? So this one cannot diffuse across the selectively permeable membrane. Then we have iodine and also distilled water. Now, iodine and distilled water, they are small molecule. They can diffuse across. This one can diffuse across. Can diffuse because they are small molecule. Okay, so now this is the one thing that we have to understand first. Iodine can diffuse across, starch cannot diffuse across. Usually inside your beaker is your starch. Oh, sorry, inside your Visking tubing is your starch. Because if you put your starch outside, right, it doesn't make sense. So it has to be inside, guys, because if it's outside, it doesn't make sense. So when you tie the Visking tubing, again, you go and watch the video of how I tie the Visking tube. When you tie the Visking tube, you can put the one that you didn't open the area, okay, at the bottom. The one that you just tie, you put it on top, and then you just add another thread to add on with the glass rod. So please go and watch how to tie a Visking tube first before you come to this question. Okay, it's like a 4-minute plus video. Go and watch the video first. Go to my Instagram, watch the video first, because that is the most important step. Okay, the most important step is know how to tie a Visking tube. Okay, the following is experimental procedure. Tie tightly one end of the Visking tubing with a 15 cm thread. Measure 8 ml of starch suspension by using 10 ml of syringe. Fill Visking tubing with starch suspension. Record the color of suspension in table 1. Tie tightly to the other end of Visking tubing by 15 cm thread. Rinse the outer surface of Visking tubing with distilled water. Tie one of the end with Visking tubing to a glass rod using 20 cm thread. Place the Visking tubing that has been tied to a glass rod into empty beaker as shown in diagram 1.2. So this is how it looks like. You can tie it and you can just add the solution inside or just tie it over here. In this procedure, I haven't finished yet, but in these few sentences, we have to find out which one is the precautionary step. The precautionary step on this experiment, there's only two. First, they are referring to tightly. So tightly is the precautionary step, which is number 4. Rinse around at the Visking tubing is also the precautionary step. So these two are the precautionary step. Remember, is tight tightly. You tight tightly, and the other one is rinse on the outer layer. Okay, why you have to rinse at the outer layer of the Visking tubing? Because when you use a syringe, sometimes you don't aim properly, so you go outside of the Visking tubing. So that's why you have to rinse around the Visking tubing before you immerse into the distilled water again. Okay, 8. Measure 250 ml of distilled water by using 100 ml of measuring cylinder and pour into 250 ml of beaker. Measure 5 ml of 1% of iodine solution by using a 10 ml measuring cylinder and add to a beaker containing the distilled water. Stir the mixture by using a glass rod. Observe the initial color of the mixture in beaker. Record your observation in table 1. Place the Visking tubing into a beaker containing mixture of distilled water and iodine solution. Make sure the Visking tubing is fully immersed into the mixture as shown in 1.1. Start the stopwatch. After 10 minutes, observe the final color of the starch suspension in the Visking tubing and the mixture in the beaker. Record the observation in table 1. Based on your experiment that has been carried out, answer the following question. Identify responding variable, because we have MV, RV, and constant. Responding variable, we have many of it, but maybe you can try to find which one is your manipulated variable. Your manipulated variable, there's only one. We call this as size of molecule. This is your manipulated variable, size of molecule. Responding variable, we have a lot of it. We have form. Final color of content in Visking tubing or beaker, color changes in Visking tubing or beaker, presence of molecule in Visking tubing or beaker, ability to diffuse across Visking tubing or selectively permeable membrane. Usually for me, I will choose this to write in class. I usually write this in class, which is presence of molecule in Visking tubing and beaker. You can choose either one, guys. It's up to you. It's your choice to write whichever one, because whichever one here is correct. You just choose one. Presence of molecule in Visking tubing or beaker. This is the correct answer also. If you want to write color also can. If you want to write selectively permeable also correct. Constant variable and method to handle. When it comes to method to handle, for constant variable, we have a way of answering. Now, how do you answer method to handle? For constant variable, we have to write the word fix. Fix constant variable at what value by using what apparatus? Okay, now we're going to find the constant in the procedure just now. Okay, in the question just now, I'm going to find which one is the constant. So what is the constant right now? 8 ml of 5% of starch suspension. This is the constant. Okay, and then we also have what? We also have here 250 ml of distilled water is also constant. 5 ml of 1% of iodine solution is also constant. Time taken is also constant. So it's up to you to choose either one. You want to write the length of Visking tubing also correct. But in the question over here, right, you don't really see length of Visking tubing because they didn't give you the length of Visking tubing, right? And it's over here. They didn't give you the length here. Unless they give you the length of Visking tubing. But if it's not, if they didn't give it to you, then we don't write the length of Visking tubing. Unless they give it to you. Usually the length of Visking tubing is about 12 to 15 cm. Usually it's 12 to 15 cm. Usually lah, but if they give it to you, then you write. If it's not, you can write type of Visking tubing. You can write the length of Visking tubing also can. So how do you want to form the sentence? Again, you have to write fix constant, fix the length of Visking tubing example at 15 cm by using meter rule. You can also write fix the time taken of immersing Visking tubing into beaker at 10 minutes by using stopwatch. So again, we have a lot of constant variable that you can write. So this is the constant that you can write. Again, how to write? Constant variable, this is your answer. Any answer also here also correct. Method to handle, remember to write what? Fix constant variable. Okay, this is your constant variable. This is also your constant variable. At what value? Value. But because concentration, we don't have apparatus. Length of Visking tubing, we can also write fix the same length of Visking tubing at 10 cm. The value, this one is your constant variable. So usually we have to copy the constant, copy inside. Your constant has to copy from the constant, yeah? If you want to write the 10 cm, you can say by using meter rule, by using ruler also can. Over here, you can write by using ruler. Okay, then duration is the same as well. If it's a duration, you can write time to immerse into Visking tubing of distilled water and 1% of iodine solution, or you can say Duration also can, time also can. Means slash is either one, whichever the word that you want to write. Remember to write what? Fix. Fix the same time. Immersing into solution X, which is per minute. Okay, this is constant and this is the value. So again, it's the same, guys. Whichever one. Don't need to memorize everything one, because in exam, maybe they only ask you to write one or two. Whichever one or two, because every experiment is going to be the same. You can write the time duration. You can also write the length. You can also write the type of Visking tubing. Up to you. You just have to change the concentration or the volume only. Up to you, okay? Your choice to write whichever one that you want. Okay, so this is how do you write. Method to handle constant variables. Okay, how do you want to write method to handle responding variable then? If it's a responding variable, method to handle, maybe I write it at the top lah. Okay, method to handle. Okay, method to handle manipulated variable. We have to write use different MV. Okay, which are what example? Okay, which are what example? Maybe use different solution, or use different size of molecules such as starch and iodine. This is the example. Okay, now the method to handle for responding variable. We have to say observe and record. Observe and record your responding variable. Observe and record. That will be your, where there is no measure. It's only observe and record, because in the entire paper 3, we only have 12 different questions. They will ask 12 different questions. So let's say if it is not osmometer, which I don't think is an osmometer, we only left 11 questions. Why? Because this experiment, we have no graph. No graph means minus one type of question. We left 11 type of question. The 11 type of question, if it is a guided experiment, then it will be 10 questions. Only left 10 questions. Whether to know whether it's a guided or unguided, because biology is on the Wednesday, right? I mean, because the one when I record this video is not Monday and Tuesday, it's like way before. Okay, so if you go for exam already for Monday and Tuesday and you're watching this video, right? If your Monday physics is guided, okay, chemistry is unguided, high chances biology will be guided. Out of 3, there will be 1 unguided. If your first subject is unguided, which is your physics, chemistry and bio, it will be guided. Usually it's only 1 unguided lah, unless you are the luckier. 3 also unguided, then nobody can help you. Then you have to memorize the procedure. But it's okay, you have time, guys, because after your chemistry exam, you have the entire day for you to revise the entire experiment again. So just remember the procedure. Not very difficult to remember the procedure also, actually. Later, I'll give you how to write a procedure later on. Okay, now this is how we write the method to handle for MV, RV, and CV. Okay, next question. We have to learn how to write hypothesis. Now, hypothesis, right? In this case, hypothesis always need MV and RV. But this paper 3 experiment, you cannot write the higher the MV, the higher the RV, because there is no value in your MV and RV. It's the size of molecule. It's not value, guys. You cannot the higher and the higher. There's no value over here. So we have to write which one is small, which one is big. Which one can diffuse across the Visking tubing, which one cannot diffuse across the Visking tubing. You still have to involve your MV and RV. If you don't know what to write in an exam, memorize this and go in and write, guys. Memorize this, you go in and write. You should get a full marks for your hypothesis. Hypothesis can be 2 marks, it can be 3 marks. So if you read the sentence, right, you can see, small molecule able to diffuse through selectively permeable membrane. Many students, they end here. The moment you end here, you never get full mark because you never write large molecule. So you have to write the large molecule. So you see, right? The small molecule is your MV. Okay, diffuse across the selectively permeable membrane is your RV, because as I mentioned, responding variable, we have a lot of it. Okay, and then same goes to this, large molecule is your MV, and then cannot diffuse is your explanation, right? We have MV and RV. Able and unable is the one that you have to write in this hypothesis. Who can pass through? Who cannot pass through? Everything needs to involve MV and RV. Everything, okay, you have to involve MV and RV, yeah? Then we have to record the result of the experiment. They ask you to record initial and final color. Now, how do I know which one is initial and final color? Visking tubing, inside contain of starch. Okay, now think about it again. Just like I show you, right? Visking tubing, we have starch. Obviously, starch cannot go out. But what do we have outside? Outside, we have iodine solution. Can iodine solution go inside the Visking tubing? The answer is yes. But they ask you to write initial and they ask you to write the final experiment, the end of the experiment, right? So the initial in the Visking tubing, the starch suspension is colorless. Just now we learned, right? It's colorless. So the initial color is colorless. If it's a final color, okay, it will be blue-black. Why? Starch is present in the Visking tubing. When you do an iodine test, it will be yellowish brown to blue-black solution. This is the blue-black solution. I would suggest you to write solution at the back lah. So write blue-black solution are better lah, because maybe in some of the exam, the teacher want to be exactly solution. So you write blue-black solution. In the beaker, what do we have? Distilled water and iodine solution. We know that starch cannot diffuse across the Visking tubing. So initially, the color is yellowish brown. Why is it yellowish brown? Because iodine solution is yellowish brown. Final color is still yellowish brown. Why? Because starch cannot come out from the Visking tubing. The color remains yellowish brown. So when you write yellowish brown in the initial color, you cannot write remains the same. Remains the same is not the answer that you write for this observation. Guys, it's an observation. They don't know what is, the teacher don't know what is remains the same one. You have to write the color itself, which is yellowish brown color. Now, based on the results, state an inference for an observation. How to write inference? Inference, you have to write RV, MV, sorry, maybe the other way around. MV, RV, reason. Your manipulated variable, responding variable, and your reason. Your reason is very simple. Whether they are large molecule or small molecule, that will be the reason. Entire experiment is just reasoning, whether they are large or small molecule, whether they can diffuse across the selectively permeable membrane or not. So in the Visking tubing, we know that iodine can diffuse across, right? Why? Because iodine is a small molecule. So the first answer that you write, iodine molecule is small. So iodine is your MV because it's a size of molecule, right? Small is your reason. They can diffuse across the Visking tubing is your RV. Either you want to write Visking tubing or you want to write selectively permeable, up to you lah. In the beaker, the other way around. Just now you say that iodine is a small molecule, right? So starch is a large molecule. Starch is your MV. The reason is they are large. They cannot diffuse across the selectively permeable membrane. So one is can diffuse across, one is cannot diffuse across. Can you see the answer is actually the same? But this one you have to write the reason, which is whether they are large molecule or small molecule. This is how we write inference. Again, if you don't remember, just remember the answer, go in and write the answer, okay? Next, what is the similarity between the Visking tubing and plasma membrane? They ask you to write only 1 mark. Don't need to explain your answer. Just write they are selectively permeable membrane. Selectively permeable means they will only allow certain substances to pass through. If they ask you to explain, then you write only allow certain substances to pass through. They will be your selectively permeable membrane explanation. If you have to write similarity, I would suggest you to write both are, write both the word both. Both are selectively permeable. Because in textbook they write selectively permeable, right? So I would suggest you to write selectively permeable instead of semi-permeable. Okay, based on the result of the experiment, arrange the following list in the ascending order. Ascending order means what? Ascending means small to big. Okay, small to big, it's called ascending. The one that can diffuse across the Visking tubing, it has to be the smallest, which is iodine. Then the pore of Visking tubing, then the starch molecule. So we follow this sequence. When you want to write in sequence, we use arrow instead of comma. Some teacher, they're very like specific over this comma and arrow kind of thing. So we use arrow instead of comma. Okay, we use arrow instead of comma. So we follow small to big. If it's descending, means big to smaller. Okay, next. Experiment is repeated using the setup of apparatus in 1.3. Benedict test is conducted on the solution in the beaker after 30 minutes. They conduct what? They conduct Benedict test. Okay, Benedict test. They use fructose instead of starch. Okay, they use fructose instead of starch. So fructose is a reducing sugar, right? Just now we learned fructose is a reducing sugar. What is the original color for Benedict test? It's a blue color, right? So blue, they will turn into brick red precipitate. Why? Because we learned that the Benedict test, precipitate, okay? Because we learned that Benedict test is a copper 2 sulfate. They want to form into copper 1 ion, copper 2 into copper 1 ion, right? Since we know fructose is a reducing sugar, can fructose diffuse across the selectively permeable membrane of Visking tubing? The answer is yes, they can, because they are small. Fructose, glucose, galactose, three of these things that can diffuse across it because they are monosaccharides. Mono means only one sugar. So they ask you, predict the result. The result turn into brick precipitate. Blue solution turn into brick precipitate. They ask you to explain why. Then you have to say, fructose is a reducing sugar. Or you can say fructose is a small molecule. Fructose is a monosaccharide. They can diffuse across the Visking tubing, whichever one also correct. You can just write fructose is a reducing sugar. Or you want to base on the size, you can say fructose is a small molecule that can diffuse across the Visking tubing. Okay, next, what would happen to the Visking tube if the solution in the Visking tube is replaced with a saturated sucrose solution and left it for an hour? The question is saying what? What happened to who? Visking tube. They never ask about the color. They ask you what happened to the Visking tube. Now, for a saturated sucrose solution, saturated means what? It means they are hypertonic. Makes sense, right? Saturated means they are hypertonic. Okay, saturated means they are hypertonic. So over here, we replace this up. We write over here. This is saturated sucrose solution. And then outside is still distilled water. So we know that this is hypertonic. The distilled water is hypotonic. Water always diffuse from more water to less water. Hypo means what? Hypo means more water. Hyper means lesser water. Water diffuses into the Visking tubing. What happened to the Visking tube? Visking tube will expand. It becomes bigger in size, expand and becomes turgid. Why? Because we have to say saturated sucrose is hypertonic than distilled water. Water diffuses into the Visking tubing by osmosis. Okay, this is the answer here, where you can say Visking tubing will expand and becomes turgid. Saturated solution has a lower concentration of water molecule than distilled water. You can say hypertonic. Instead of this, you can write hypertonic lah. You can write hypertonic also can. Water will enter to the Visking tubing through osmosis. You have to write osmosis. The Hitch question is actually a prediction question. Again, as I mentioned, right, we have 11 questions left. One of the questions, it will be prediction. Prediction question, it can be a lot of things. Usually it's within chapter 3. They ask you a hyper, they ask you about what happened when you replace the solution with the sucrose solution, replace with other solutions. So it's a prediction. You predict it. Even if the question in the Visking tubing, you don't put glucose. In the question itself, you will still see glucose. Like for example, this one. In the question just now, when you do the experiment, you have no glucose, right? But in the question over here is a prediction question. They will still give you glucose, which is a reducing sugar. So you still have to understand when there is a glucose and when there is a starch. What is the color of it? There's only 4 colors that we have to know. Yellowish brown, blue-black solution, blue to brick precipitate, of course, colorless. There's only few colors that you can write. You cannot write rainbow color, right? You cannot write purple color, right? Because there's no such color that we learn in this part. Okay, now, this is first type of question. Then we're going to do second type of question. And the reason why I'm showing the answer is because that I want to be fast. So later you can stop the video and write the answer by yourself. Okay, now, second question. Same thing, they give you a checklist and then you check your answer. Remember to utilize your time, yeah? If you're soaking your distilled water, if you're soaking your Visking tubing, then you just try to do the question straight away. Don't wait for the time to pass. Utilize your time. If you cannot tie the Visking tubing within 20 minutes, then forget about tying the Visking tubing. Don't tie already, guys. Straight away, you do your experiment. Straight away, do your paper. Don't waste your time tying already. Okay, you are required to carry out. This is carry out. You are required to carry out an experiment to study the movement of substances across a selectively permeable membrane based on different size of molecule. Diagram 1 shows the setup of this experiment. Plan this experiment by using apparatus and material provided. Your procedure should include method to handle MV, method to handle LV, and precautionary step. As I mentioned, right, how do you explain this kind of question? Method to handle for MV, you have to say use different. RV, you have to say observe and record. Precautionary step, there's only 2, which it will be tie tightly and also rinse the Visking tube around it. You have to rinse around because you want to prevent it. So you rinse around it. So we have the criteria over here, categorized in color for you already, so you can look at the color itself. So the one in red color is a general answer. Sometimes it's just your constant variables because they didn't ask you to write constant, right? So it will be constant variable. So for P1, prepare a Visking tubing at 15 cm in length. This one is your constant variable. maybe I use pink color, okay? This is your constant variable. Write it over. This is your constant variable. Because the question didn't ask you, but we still write it lah. P2, tie one end of Visking tubing by a thread tightly. Tightly is one of your precautionary steps. Fill 15 ml of solution P into Visking tubing, which is your MV, because your solution P right now is the size of molecule, right? Is your MV over here. Tie the other end of Visking tubing tightly. Again, tightly is your precautionary step. And rinse the outer layer of Visking tubing by distilled water. Fill 150 ml of distilled water in beaker. This is also your constant variable for P6. Fill 10 ml of solution Q. This is also your MV, manipulated variables. Immerse the Visking tubing into beaker. Record the initial color of the solution inside the Visking tubing and outside of the Visking tubing. Start the stopwatch and wait for 15 minutes. This is also the constant variables. Your responding variable is after 15 minutes, record the color of solution inside Visking tubing and outside Visking tubing. Record all observation in table. This is what you have to write in exam. I know the question is only 3 marks. Sometimes in your brain, right, you cannot brain which one to write or not to write. Just write anything, everything. If your first exam is unguided, then it's okay. You can just forget about the procedure because I don't think they will ask you this lah. But it's okay, guys. There's only one procedure that you have to remember, which is this one. So remember this, guys. Okay, go and remember this. Please, okay, please you all go and remember this. It's only one lah, okay? Make some time to remember the procedure. Don't just wait for nothing. It's a 15 over 15 marks. Do you want to get full marks? Of course, you want to get full marks, right? Now, I already give you the question. I give you the possible question that you will see in your exam also. You only have to do one thing. Remember, understand the concept. Since we have solution P and solution Q, we don't know what is it. But I can tell you whatever is inside your Visking tube, it will be your starch. Because as I mentioned, right, if you starch you put outside, right, it doesn't make sense, one. Because if you put your starch outside, then what is the purpose of doing this? Because starch cannot go in, but iodine can go out. Means you have no result. So usually we put starch inside the Visking tube. Usually lah, I would say usually lah. Solution Q, it will be your iodine. Iodine solution. So P is starch and Q is iodine. So iodine can go into the solution P. Okay, next. Based on the experiments, state the color changes occurs in the Visking tubing after 15 minutes. Iodine, can they diffuse across it? We say that iodine is a small molecule. So iodine can diffuse across it. Original color of iodine is yellowish brown color. When after the experiment, since there is starch inside, so the yellowish brown, it will turn into blue-black solution or dark blue solution also can, blue-black or dark blue, but it cannot be blue or black. Explain your observation. Again, how do you explain your answer? Explanation always right, whether they can or cannot diffuse across the Visking tube. Okay, you see, right? Substance Q, which is your iodine. Iodine can pass through the Visking tubing, or you can write molecule Q present in the Visking tubing, whichever one. But usually I will write the first one now. Molecule Q, it can still present in the Visking tubing because the size of molecule is small. This is your observation. Usually your observation also have to include your MV and RV because it's what you see. Don't have to use a lot of brain cell because it's just what you see. If it's not, you can just write molecule Q still present in the Visking tube. It's up to you, whichever one. Okay, whichever, because the answer is the same. Construct a table to record the result of your experiment based on the criteria. In question, they will give you what you need. They ask you to write this in your exam. Color of solution, in the beaker, in the Visking tubing, observation, beginning and also the end of the experiment. So when you write, you draw accordingly. No matter how you draw, doesn't matter. As long as you're able to involve this in the table. This is the table that you can see. How do we get marked over here? You get marked by writing, number 1, the labeling at the side. The labeling over here, number 2. Number 3 is your result. Number 4 is your result as well. So in the Visking tubing, what do we have over here? In the Visking tubing, before it will be cloudy solution. Either you write cloudy or either you write colorless, up to you. Cloudy or colorless, both also correct. At the end of the experiment, you get a blue-black solution. Why? Because the iodine can diffuse into the Visking tubing, right? So you get a blue-black solution. Then we have outside. Because we have iodine in the Visking tubing, it will be yellowish brown color. At the end, the starch cannot come out, right? It's still yellowish brown color. So this is how we draw. It's also a mark that you can get it easily as long as your result is correct. Even your result is wrong, you still get 2 marks by writing the title. You still get 2 marks over here. Okay, next question they ask. Based on the result in the experiment, classify substances based on the size in table 1. So we have the small size and large size. So we know that iodine is small. So we know that Q is a small. So Q is small, P is large. Don't write iodine, don't write starch. You copy the question. The question give you Q and P. You write Q and P, guys. Do not write starch and iodine, because what if you write it wrongly? Okay, what if the question is not starch and iodine? Who knows, right? Then you copy from the question. Everything you can copy from the question one. You don't have to write starch and iodine. A pupil repeat this experiment by replacing a solution P with juice X in the Visking tubing. So you're still doing iodine test, but you're doing with what? You're doing with a sucrose. Juice X, this is a sugar cane. Sugar cane is a sucrose. Can sucrose react with iodine? Of course, the answer is cannot. Sucrose cannot react with iodine. And for sucrose, usually we will do Benedict test. The result will still remain as blue color. If you want to hydrolyze, if you want your sucrose to have a positive test in Benedict solution, you have two methods. Number one, you add with diluted hydrochloric acid to hydrolyze it. Number two, you add enzyme to hydrolyze it. Then you're going to have a break with precipitate. If it's not, your sucrose will still remain as blue in Benedict solution. Obviously, right now, it's not even Benedict, it's iodine solution. Iodine solution, the iodine will always remain as iodine color, which is what? Yellowish brown color. You can say no changes in color, but if it's me, I will write no changes in color, which remains as yellowish brown color. So I will usually write the color out, because sometimes the teacher wants you to be very specific about what is the color. So you can just write no changes in color, which remains as yellowish brown color. They ask you to explain why. Because sugar cane is sucrose. Sucrose cannot react with any iodine, so it remains the same. So you're going to say just X is sucrose. Sucrose is a disaccharide. They do not react with iodine, so the result remains the same. So there's one more question I didn't include over here. Is actually the operational definition. Okay, now how to write operational definition? Operational definition. Operational definition. Definition. They will usually ask about selectively permeable membrane. Selectively permeable membrane. They ask you, what is a selectively permeable membrane? Now, how do we explain this operational definition? Definition, no wonder. Keep on thinking the spelling is a bit weird. Okay, definition. Now, how do we want to write operational definition? We have a method of answering. We have to write theory, which is shown by RV. RV affected by MV. Okay, RV affected by MV. Theory, which is shown by RV affected by MV. What is the theory of selectively permeable? It only allows certain substances to pasteurize. How do we start our sentence? Usually you have to start with selectively. This is your answer, yeah? Selectively permeable membrane is a membrane that allows certain substances, or you can write small molecule, to move freely across it. Okay, this is your theory. You're going to say, which is shown by, what is your RV just now? We write RV is what? The presence of molecule in Visking tubing and beaker affected by, what is your MB? Size of molecule. So what do you have here? You have theory. This is your theorem. Theory, which is shown by RV affected by MV. So in this video, I've taught you 12 different questions that you will see in your exam. But besides prediction, because prediction sometimes is a bit hard to predict what they're going to come out. But at least you know the rest of 11 questions. You know how to write hypothesis, your MVRV, your method to handle, your table. There's no graph here. So 11 questions. Okay, you know how to write all of the question already. So hopefully that in your exam, you can score a full mark. Okay. I hope that you all can get 15 over 15. I hope that you all learn something from this video itself. So if you have any question, maybe you can comment at the comment box. If I see, maybe I will reply, or maybe your friend will reply on behalf of me as well. Okay, so hope you guys have fun learning this. Good luck for exam, guys. Bye bye. See you guys next time.